Hello, my name is Deng Luo. I'm a PhD student at Chaos Analysis Group. In the following half hour or so, I will present the structural biology background of Bacteriophage T4 atomistic model that we built. This slide shows one mutant of T4 called isometric T4. The left and the middle are the same models with different color schemes, while the rightmost one is the retracted configuration that is unfavorable for infection. Those models are created by an in-house tool developed here in Chaos Analysis Group called MesoCraft. Shows on the upper right is the graphics window of it. It uses simple rules to allow modelers to rapidly model mesoscale biological models. Lower right shows, lower left shows after you specify the so-called siblings rule between two instances of the same protein, then a line of this type of protein will be automatically populated once you apply the rule. And on the middle, it shows the result of creating a lipid monolayer patch. On the right, it shows the SARS-CoV-2 model created by MesoCraft. This tool is published in this year's IEEE TVCG. To make it concise, for all the references, I will write only the first author and the year followed by a ref number, which can be used to find the detailed reference in the end of the presentation. This is just a quick demo of what MesoCraft can do. However, in this presentation, I will focus only on the biology part of T4 model, basically how the model should look like rather than how it is created or what rules it uses. We will have follow-up materials to cover the latter. So here's the overview of T4's structure. It can be divided into three parts: the head capsid, the tail that has the tube and base plate, the fibers, the long tail fibers, and the wac-collar and whiskers. This page shows the head capsid conformation. It is an elongated icosahedron. GP23, which means gene product 23, forms the hexagonal capsid lattice, which is this yellow greenish protein. And GP24 forms pentamers at 11 out of 12 of the vertices of the icosahedron, while GP20 forms the unique dodecameric portal vertex. And every GP SOC molecule binds between two GP23 subunits. That's here. Every GP SOC molecule binds between two GP23 subunits, but not between GP23 and GP24. So here we don't have SOC protein. And GP SOC forms trimers at the meeting points, here at the meeting points of three GP23 hexamers. That's this three. And also SOC attaches only to the surface of the capsid. And in this slide, we shows the triangular number of the head capsid. The wild type T4 has two triangulation numbers, T equals 13 and T equals 20. T equals 13 covers the top and the bottom cap, while T equals 20 is the middle section. And there is a mutant of T4 called isometric, and that has T equals 13 for all the faces. Sorry, if I pronounce it wrongly, it should be T equals 13 for the cap and the bottom of, for the top and the bottom cap of the wild type prolate T4. Layman wrote in 2003 that for isometrics, the head diameter was found to vary from 973 angstrom along the 5-fold axis to 879 angstrom along the 3-fold and 2-fold axis. While for the wild type, the variant consists of 1150 angstrom long and 850 angstrom wide prolate head with hammy icosahedron ends. However, Rao wrote in 2010 that the capsid is 120 nanometers long and 86 nanometers wide. So in our modeling, we used the following parameters and found it fits well with the PDB structures we are using. The height is defined as the distance between the top and the bottom vertices. It is 86 nanometers for isometric. The width is defined as the widest vertices between the in the pentagon plane of this one or this one, and it is 74 for isometric icosahedron, and the diameter is the same as height. It's 86 nanometers in this type of icosahedron, while for the elongated wild type, the height is 115 nanometers with the same width as the isometric icosahedron. So that gives an equivalent diameter of 86 nanometers. So now we start looking for the building blocks of the head. We use experimentally solved structures in the PDB database whenever possible. For GB23, we found 5VF3. It contains several other proteins as well. We isolated a hexamer out of it for GB23, and then used the hexamer to build the capsid. The final capsid has 6, 1, 2, 3, 4, 5, 6 hexamer for each face, and in total 120 hexamers. And that is equal to 720 GB23 monomers. And here on the lower right shows the result. This is a cutaway view, and this is the overall view. We don't use this complex even if it is the T4 isometric capsid directly, because it lacks GP20, but it already forms a icosahedron. So there is no space for GP20 to be fit in. But we will use the orientations of various proteins inside this complex as a reference when we build the capsid. We isolate GP20 monomer from PDB ID 6UZC and arrange 12 of it in the same way as shown in this PDB at the bottom vertex of the icosahedron. Here shows the result, and this the left shows it in relation to GB23, while this right shows the position on the icosahedron skeleton. In total, there are 12 GB20. We use PDB ID 1YUE for GP24 monomer and build a pentamer for each of the rest 11 vertices. The pentamer's orientation is aligned to that in the 5VF3 complexes shown just now. SOC is isolated from this big complex again. We use the 11mer to build the capsid to keep the subtle orientation details the same as that in the complex. In total, 60 of these 11mers are used. So there are in total 660 SOC. The only Hawk structure information is found in this complex again, but it has only 25 amino acid being solved. So we are using the AlphaFold predicted result. Hawk is placed in the middle of each 20 GP23 hexamers. So in total, there are 6 multiplied by 20. That's 120 Hawk. If we place the Hawk in the same placement as that in this big 5VF3 complex, overall on the capsid, it shows as an irregular shape, and it does not resonate with what Lyman wrote in 2003, that up to 2/3 of GP Hawk might be inserted in the center of each GP23 hexamer. So we then place the Hawk along with the normal of the capsid and has 2/3 of it inside the capsid and put the ball-like shape outside. So by now, we have the full head for the isometric T4. This is a comparison between the model and the EM image. It looks almost the same. Now we move to the tail. At first look, the tail is really stunning and intimidating to model, as it contains many different building blocks, as shown on the left figure. However, the base plate that has these many proteins is solved in one complex called 5IV5. So we just use this complex, but separate the different proteins so that we can color them differently in MesoCraft. The so-called tube protein GP19, we used 5W5F, and we use 3J2M for the sheath protein GP18, that is here, and the sheath terminator protein called GP15. The result is shown here on the right. For the sheath, our model reflects what Lyman wrote, that the subunits comprising each cylinder form a 6-star helix. That's this 6-star helix with a pitch of 41 angstrom and a right-handed twist angle of 17 degrees. The helix has a length of 984 angstrom. Lyman wrote also it contains 24 repeats, basically 24 layers of the GP18 and GP19. However, we use this most recent information from YAP in 2014. This one in this diagram, it has 138 GP19, the same for GP18. That is 23 multiplied by 6. So we put only 23 layers of the sheath and the tube hexamers. However, it shows here there is GP26, GP28, and GP51 in the base plate as well, but those have too little information, both on the literature and also on the structural wide. So we therefore do not include them in our model. Immediately above the tube protein GP19 here is not the GP15, but the so-called tube terminator protein, that is GP3. And unfortunately, we don't have the structure information being solved. So I used AlphaFold to predict. And there is no further evidence other than the copy number is 6 from Lehman in 2003. So we placed a hexamer above GP19 at a reasonable volume occupation out of our imagination. So shown here, the yellow is 18, the red is the tube protein 19, and this purple is 3. Then there are GP13 and GP14. That's called the head compilation proteins. Between the tail and the capsid, the information about them is even more limited, that the copy numbers are not clear, let alone the soft structures. So I used AlphaFold again to predict both of them. We then formed a ring. We then built a ring formed by 12 of GP13, and then surround the ring with 6 of GP14. So why is that? Here in this slide, Achter wrote in 2007 that in vitro, they observed a 5-fold configuration, which has, for each fold, it has 2 GP13. And this is GP13 is in circle, and 1 GP14. And in 5-fold, in total, that has 10, 13, and 5, 14. However, Lehman observed in 2004 a 6-fold configuration, but they concluded this only tentatively because they lack the structural information and the resolution is not that high. We went with the 6-fold version because, first of all, the 5-fold result was from in vitro, and all the other proteins like the this tube and the sheath, and also the terminators, the sheath terminator and the tube terminator are all in 6-fold configuration. So we went with 6-fold here. Then there is the so-called tape measure protein, GP29. Fokine drew in a diagram in 2014 that it just drops in the middle of the tube. We first used AlphaFold to predict the structure, and then you can see here it all stays in nearby for the alpha helix. So I then manually refined this structure in PyMo in its editing mode by moving the segments to make it almost a straight line. We then placed the N-terminal close to the head capsid because we cannot find the orientation information. So we just choose one. By now, we have the head capsid and the tail. Now we move to the fibers. The neck first. Neck fiber is composed by a protein called WAC. And we used PDB ID 2BSG for it. It has 2 configurations. 6 of it forms the collar. That's this horizontal, that's in this horizontal plane. And the other 6 form the whiskers vertically. For the whiskers, we based our model on this image shown here. we made the WAC here, those WAC touching the GP14 that goes inside this ring. But the placement of the collar WAC, again, is somewhat out of imagination. And we believe that this brown densities here is unassigned densities by then. And we believe those are the collar composed of the WAC proteins. So we placed the collar and the whiskers and made the outlook the same as the figure from all of these three view directions. So this one is close to this view, this is close to the lower left, and this one is when you view from the top. It's almost identical to this result. Finally, we move to the legs, formerly called as long tail fiber. I will now walk you through this big chunk of text while drawing on the figures to make it less boring. So LTF has kinked structures of about 1,440 angstrom long. That means this distance is around 144 nanometers with a variable width of up to about 50 angstrom. That means this length is about 5 nanometers. And LTF can be divided to proximal, that's here, proximal and distal half fibers attached at an angle of about 20 degrees. And we think it means this degree is 20. And in the intact phage, the N-terminal end of GP24 is attached to GP9. So here it attaches to GP9, and this is N-terminal. However, GP35 and 36 is their orientations are not mentioned. And GP37's C-terminal is at the distal part and has the receptor recognizing tip. That's here, which means this side is C-terminal, and which all of this above information is consistent with this figure shown by Heyman in 2018, that this is N-terminal for 34. This is C-terminal, and then for 37, N-terminal and C-terminal. The long tail fiber is completed when the amylo terminal end of distal half fiber, amylo end, that means, and the carboxyl terminal end of the proximal half fiber complexes are attached through a monomer of 35. So that means this side is C-terminal attached to here, and this side is amylo terminal. So that confirms this N terminal should be here. So the information is consistent between these several papers. The final information is the phage proximal rod is formed by a homotrimer of GP34. So that means this rod is a trimer. And here is a monomer of GP35. And then the distal end consists of two protein trimers. So that means 36 and 37 are both trimers. Okay, then GP34 is only partially solved at C-terminal in 5NXH. GP37 is again partially solved at C-terminal in 2XGF. All the rest are not solved. So I first predicted all the monomer structures for 34, 36, 37, and 35 by AlphaFold. And then I realized that I should use some tricks to predict the trimer structure instead. While the trimers are being predicted, our server experienced a power outage, and I then decided to, again, just manually tweak the monomers, because even if we predict it by AlphaFold, it might not be precise enough. So then for manual tweaking, my first step is to straighten all of these monomers except for 35, because it is a monomer here. And then the second step is to duplicate them, like here 36, duplicate them into a trimer. And then for 34 and 37, I will delete the C-terminal part and then splice this N-terminal part with the experimentally solved domains for both 34 and 37. So they are kind of hybrid. The N-terminal part is predicted, while the C-terminal part is experimentally solved. So this page shows the result that is conformed to what we just walked through, the big chunk of text. And the N-terminal end of GP34 is attached to the base plate protein GP9 here. So now we have the full model of T4. So we also try to model the retracted configuration where the elbow region of each long tail fiber interacts with one whisker, and the distal part of the fibers interact with the collar. So in the end, it looks similar to this. But when I preparing my slides, I realized that there is a slight right turn for the GP34 protein here. We have not, we just straightened it up. So we may make another updated version where the 34 slightly goes to the right. So this page shows three versions of the phage. The left conforms to the 20 degrees requirement found in the literature, the middle for the retracted configuration, while the right for the commonly found images on the web, where the elbow is apparently bending more than 20 degrees. So we have tabulated all the building block proteins and the modeling details in a table. We can share this table upon request. Right now, we are building the wild type capsid on the elongated icosahedron. We are looking for information about the DNA packaging and thinking ways to model it. We are looking for suggestions and information on how to handle the inner contents inside the head. And we are also converting the model into MMCIF format that is more commonly used in the domain. And we would appreciate it very much if you can provide feedback on how to improve the model, and we will incorporate the feedback whenever possible. So here are all the references we have used. Thanks to all the researchers that have paved the way for us and also for the domain to have such a model. Finally, let me show you the heroes behind the scene. John, Paul, and Leon together modeled the head capsid. John generated the skeletons and worked with proteins on the vertices and pioneered the DNA modeling. Leon and Paul worked with proteins on faces and solved many occlusion and alignment issues. Alia worked on the tail and the fibers, and almost all the imagination part comes from her artistic mind. They are all high school students from the Coast School, TKS, and worked in this project in the so-called WISE, WISE internship program. Thanks also goes to Coast VCC, my advisor Ivan, who first initiated the idea of modeling T4 and provided enough resources and oversaw the whole project. Andre, the main developer of MesoCraft, helped train the students on how to use MesoCraft and along the way answered tons of questions. Ngang, the same as me, PhD student here in LOVs, helped in many discussions and is now converting the model into MMCIF format. And before and in the middle of modeling, we have occasionally consulted David from Scripps Research for feedback. So thank you all for making this happen, and thank you for listening.